Imagine a disease that's been a medical mystery for 150 years, a shadow passed down through families from one generation to the next. For all that time, we could really only watch it happen. But now, we've gone from just observing its tragic effects to pinpointing the single genetic typo that causes it all. So today, we're going to follow the clues and solve that mystery, piece by piece. Our story starts way back in 1872 with a doctor named George Huntington. And with just a few words, he perfectly, and I mean perfectly, captured the three cruel hallmarks of the disease. It runs in families, it ravages the mind, and it lies in wait for decades before it strikes. It was an incredibly precise observation for the time, and it basically set the stage for more than a century of scientific detective work. So, to solve this thing, we're going to start right where Dr. Huntington did, by looking at the symptoms, the outward signs that something is terribly, terribly wrong. Clinically, the disease shows up as this tragic triad. First up, you have the motor symptoms. This includes those signature involuntary, almost dance-like movements called chorea, but also a crippling loss of control over simple voluntary actions. Then there's a slow cognitive decline, which just erodes the ability to plan, to reason, and to remember. And finally, the psychiatric component, which can be just as devastating. We're talking profound anxiety, depression, and major personality changes. It's this cruel combination that robs people of their movement, their thoughts, and in many ways, themselves. Okay, now here is our first major clue. When you look at who gets this disease, a really stark pattern emerges. In the UK, the prevalence is over 12 people for every 100,000. But look at Japan. It's less than 1. This huge difference isn't random. It's a massive clue pointing directly at ancestry and ultimately at our genes. So, with the symptoms laid out and a powerful genetic clue in hand, the investigation has to move inward. To really understand what's happening, we need to go to the scene of the crime, inside the human brain. The epicenter of all this damage is a set of structures deep inside the brain called the basal ganglia. You can think of this area as the brain's air traffic control for movement, learning, and even emotion. It's the mission control center for so much of what we do, and in Huntington's, it is under direct and relentless attack. So, how does this control system actually work? Well, think of it like traffic lights for your body's movements. The basal ganglia uses two main circuits: a direct pathway that acts like a green light, basically saying go, and an indirect pathway that's the red light, saying stop. In the early stages of Huntington's, that red light pathway starts to degenerate first. The stop signal breaks down, and that leads to those uncontrolled, involuntary movements. But later on, the green light pathway also fails, causing rigidity and slowness. The entire system just breaks down. And if we zoom in even further, we find the specific victim. The cells being completely wiped out are called medium spiny neurons, or MSNs for short. They're the real workhorses in this part of the brain, and the devastation is almost total. Over the course of the disease, more than 90% of these vital neurons are lost forever. Okay, so we have the symptoms, we know the location in the brain, and we've identified the specific cells that are dying. Now it's time to zoom into the molecular level and find the ultimate culprit, the real perpetrator. And here it is. After decades of searching, in 1993, scientists finally found the smoking gun. And it wasn't some complex web of environmental factors or lifestyle choices. No, it was a single typo in a single gene, known as the Huntington gene, located right there on chromosome 4. The whole devastating disease written in our DNA. So what exactly is this typo? Well, you can think of it like a genetic stutter. Inside the Huntington gene, there's a section where the 3 DNA letters, CAG, are repeated over and over and over again. Now, in most people, this section is short and stable, but in Huntington's, the gene kind of stutters during replication, and this section gets copied way too many times, becoming dangerously long. Let's look at the numbers, because this is where it gets really clear. If you have 26 or fewer of these KAG repeats, you are in the clear. You will not develop Huntington's, and you won't pass it on. This is the normal, stable range. Then there's this intermediate zone. If you have between 36 and 39 repeats, you're in a gray area of what scientists call reduced penetrance. A person with this number of repeats may or may not develop symptoms in their lifetime. It's the genetic tipping point. But once you cross this threshold, once you hit 40 repeats or more, the outcome is brutally certain. Anyone who inherits a gene with this many repeats will, if they live long enough, develop Huntington's disease. period. And what's particularly cruel is that this genetic stutter can actually get worse as it's passed down, which means the disease often appears earlier and more severely in each new generation. So, finding the gene was a monumental breakthrough, absolutely game-changing, but it immediately opened up a brand new mystery. We found the typo in the instruction manual, but how exactly does that typo cause the entire system to crash? How does one faulty gene kill brain cells? Well, it all comes down to what genes do, right? They provide the blueprints for making proteins. That stuttered CAG repeat in the gene gets translated into an abnormally long, sticky tail on the Huntington protein. This mutant protein, which we call MHTT, can't fold properly. It becomes misshapen, dysfunctional, and ultimately toxic to the cell. And because they're so misshapen and sticky, these toxic proteins start to glom onto each other inside the neurons. Over time, they form these large, dense aggregates that scientists can actually see under a microscope. They're called inclusions, and they are a key hallmark of the disease. But this is where the mystery gets even deeper, and it's fascinating. We see these clumps, these inclusions, at the scene of the crime. But are they the killers, or are they something else? Hypothesis A says, yes, these big clumps are directly toxic. They physically disrupt the cell's machinery until it dies. But hypothesis B suggests something completely different. Maybe the clumps are actually a defense mechanism. Maybe the cell is bundling up all the toxic proteins to quarantine them, and the real damage is being done by smaller, invisible fragments that are still floating around. The jury is still out on this one. So, we followed the clues from a doctor's notebook all the way down to a single toxic protein. And that brings us to the most important chapter of all. What on earth can we do about it? Let's look at the hunt for a treatment. With the culprit identified, the strategy is becoming much clearer. The primary goal right now is to develop drugs that can lower the amount of that toxic mutant Huntington protein in the brain. At the same time, researchers are on the hunt for reliable biomarkers, things they can measure in the blood or spinal fluid to track the disease long before symptoms even start. And finally, by studying other genes that can delay or even speed up the onset of HD, scientists hope to find brand new ways to protect the brain. After 150 years, the fundamental why of Huntington's disease has been solved. The investigation revealed a genetic stutter that produces a toxic protein, which in turn destroys a critical part of our brain. The core mystery is gone. And that leaves us with the ultimate question. With the cause now so clearly in our sights, how long until the next great discovery is a cure?